Before we can start talking about selecting the primary antibody, we first need to determine what is our target. Now, that may seem a little obvious because by the time you're thinking about doing a Western blot, you probably already have a protein of interest. However, if you look at the pathway in which that protein plays a role, you may find out that looking at upstream and downstream proteins may allow you to broaden the pool of proteins that you can study, and that could also broaden the pool of antibodies that you may be able to use. For example, if I want to study the growth arrest pathway, I could certainly look at protein B, which is known to be a key regulator of this pathway. But I could also look at proteins A, C, or D, which directly interact with protein B. However, looking at proteins E or F may not be the best use of my time, since they're not really part of that pathway. You should always refer to peer-reviewed publications as well as online databases and other interactive resources when you are studying your protein of interest. At the end of this course, you will find a list of those resources, so be sure to take advantage of that. When you're studying your target, there's going to be 6 key pieces of information that we need to gather. First, we need to determine protein abundance. Then we're going to start thinking about what is the size of our target. We also need to find out where is that protein located in the cell as well as in the tissue if that's the case. Then we start looking at, is this protein present in different forms? We also need to take into consideration if that protein is known to undergo chemical or post-translational modifications. Last but not least, you will need to determine if you're going to obtain and handle that protein on its native state or on its denatured form. 3 of those details you're going to use to determine if your potential targets are a good fit for your particular Western blot. So grab your worksheet and let's start working through it. The first thing we want to address is how abundant is that protein. If a protein is highly abundant, it's easier to work with it because it's easier to isolate and also easier to detect it on that membrane. Second item on our list is the protein size. If a protein is bigger than 16 kilodaltons and smaller than 160 kilodaltons, give and take a little bit, they're going to be easier to work with. That's because they're easier to isolate and they're also easier to transfer when they're not too big or not too small. The third item on our list is the cellular and tissue localization. So ideally, we want to work with a protein that's present free in the cell, meaning inside the cytoplasm or inside the nucleus without it being bound to the membrane or to the DNA. We also rather work with a protein that's not restricted inside one type of organelle only and that is also not secreted. Therefore, if you can choose to work with a protein that's highly abundant, it's not too big and not too small, and that is not trapped in any particular cellular structure, that will make your Western blot protocol easier. However, if that's not an option for you, don't worry, you can still adjust your protocol.